Welcome to the deep dive. Today, we're diving into a really crucial question, I think. How do you figure out what to believe, especially when you get information that seems, well, totally contradictory?
Exactly. We've got some material shared by a listener, Elena, that really sparked this.
Right. Elaine is expecting her first child, and she's understandably finding herself in a bit of a whirlwind online,
Yeah, she's seeing articles, some pretty persuasive sounding ones, maybe suggesting a link between the MMR vaccine and autism.
which, must be worrying.
Totally. Especially when her pediatrician is giving her different information based on scientific studies, saying the vaccine is safe and effective.
It just highlights this challenge, doesn't it? Trying to make the best choice, navigating what feels like two completely different stories.
Mm-hmm.
So in this deep dive, we're going to try and unpack some of this, give you and Elena some tools maybe to sort through this kind of thing without feeling, totally overwhelmed.
We'll look at what science actually is, how scientific knowledge gets built, how it's tested.
And ultimately, how to get a better feel for what's reliable information versus maybe what's not, using Elena's situation as our guide.
Okay, so let's jump right into Elena's dilemma. She's got this online info clashing with her doctor's advice.
Yeah. And her first question from the material she sent was basically, how does the quality of that online source compare to the pediatrician's information?
Right. And to tackle that, we kind of need to talk about the foundation, the scientific method, as it's often called.
Okay, scientific method. Now, that doesn't mean like a super strict step-by-step recipe everyone follows exactly, does it?
No, not quite. It's more about a defined process for investigating things. Usually involves, careful observation, maybe doing experiments.
And testing ideas.
Exactly, testing ideas. We call those hypotheses. Think of a hypothesis as a sort of proposed explanation for something you've observed.
But the key thing is, it has to be testable.
Absolutely.
Yeah.
Testable. And these hypotheses, they usually fit into a bigger picture, what we call a scientific theory.
And a theory isn't just a guess, like, I have a theory about why my keys are missing.
No, not in science. A scientific theory is much more robust. It's an explanation that's been tested again and again, confirmed many times. It's basically our best current understanding of how something works.
Okay. And sometimes we hear about scientific laws, too.
We do, yeah. Especially in fields like physics. Laws often describe how nature behaves under specific conditions, often with a mathematical equation, like Newton's laws of motion.
But it's not like a hierarchy, is it? Like, a hypothesis becomes a theory, then graduates to being a law, meaning it's more certain. The material suggested that's not quite right.
That's a really important clarification. No, it's not a ladder of certainty. Hypotheses are what scientists work with day to day, usually within the framework of existing theories. Laws are more like concise descriptions of patterns. They aren't necessarily higher or more proven than a very well-supported theory, like, say, the theory of evolution.
Interesting distinction. So what drives all this?
Curiosity, really. Scientific inquiry. Scientists want to figure things out. And they use broadly two kinds of reasoning. Inductive reasoning.
Okay.
That's like seeing specific examples or observations and drawing a general conclusion, like noticing a pattern in brain scans when people do a certain task and concluding that brain area is involved. Lots of brain studies work like that.
Makes sense. And the other type?
Deductive reasoning. That's more like starting with a general principle or theory and predicting a specific outcome. If the theory is true, then we should see X happen in this experiment.
Gotcha. And back to hypotheses, you said testable.
No.
What else?
Crucially, they also need to be falsifiable.
Falsifiable. Okay, what does that mean exactly?
It just means there has to be some conceivable way, through an experiment or observation, to show that the hypothesis is wrong.
Ah, okay. So science isn't about proving things with absolute certainty.
Not really. It's more about finding evidence that either supports an idea or refutes it, shows it's likely incorrect. We're always testing. The material even mentioned in silico research.
Yeah, what's that?
It's basically using computers to test hypotheses now. Analyzing huge data sets, simulating things like how a potential drug might interact with a virus protein before you even go into the wet lab. It's another tool for testing.
Huh. Cool. So, thinking back to Elena.
Exactly. We need to ask, does that online article she saw present its claims in a way that reflects this process? Are there testable ideas? Could they potentially be proven wrong?
And what kind of evidence are they actually using compared to what her pediatrician is drawing on?
Which is likely based on these broad, well-tested scientific theories and lots of data.
Right. Which leads neatly into Elena's second question from the sources. Are those arguments linking MMR and autism scientifically valid? And could there be other explanations?
This hits on how science actually changes over time. It's not static.
It's not set in stone.
Definitely not. Ideas get revised, updated, refined as we learn more, as new observations come in. Usually it's not a complete throwing out of old ideas, but more like adding detail, seeing the bigger picture.
Like the example in the material about Mendel's laws of inheritance.
Yeah, exactly. Mendel figured out some basic rules back in the 1800s. They still hold true, but now we understand them within the much, much broader context of DNA, genes, chromosomes. It's a more detailed picture.
But sometimes there are bigger shifts, right? The sources call them paradigm shifts.
Yes, absolutely. Those are the more radical changes in thinking. The theory of evolution is a classic example in biology.
Going from thinking species were fixed and unchanging.
To understanding that they evolve over time through natural selection. That was a massive shift. And those big changes usually need a lot of compelling evidence, and they often face resistance at first.
Okay, so relating this back to the MMR vaccine and autism claims.
Well, that specific idea traces back primarily to one single study. A study that has since been thoroughly discredited, retracted, and found to be fraudulent by the scientific community.
Wow.
Well, the initial observations might have seemed concerning to some parents. Those observations just haven't held up when tested rigorously in many, many large-scale studies afterward.
So those later studies, the massive ones, represent what you called consensus science.
Exactly. Consensus science. That means ideas that have been researched extensively over years with tons of evidence piling up, all pointing generally in the same direction.
Like climate change, the material mentioned.
Yep. Another very clear example of scientific consensus based on decades of research from multiple fields.
And this is different from, what was it, frontier science?
Right. Frontier science is where the action is happening now. New ideas, maybe initial findings, things that haven't yet got that huge body of supporting evidence. Think about ongoing debates in nutrition, for example, about specific diets or supplements.
And the media often jumps on that frontier stuff.
Often, yeah, because it can seem more newsworthy, maybe more controversial. But that can sometimes give people the impression that all science is that uncertain or up for grabs.
Which isn't really the case when you have strong consensus.
Precisely. So for Elena, a key question about her online sources: is it reflecting this broad scientific consensus on vaccine safety? Or is it pushing an idea from the fringes, maybe one that's already been tested and refuted?
And crucially, has it been peer-reviewed? That came up in the source material, too.
Yes, peer review is so important. When scientists submit work to a reputable journal, other experts in that exact field anonymously review it.
Okay, what are they looking for?
They check if the research is original, if it's significant, if the methods are sound, if the conclusions logically follow from the data, if it's thorough. It's a critical quality control step.
A filter, basically.
A very important filter. Many sources online, like blogs or advocacy sites, just don't have that level of scrutiny. There are exceptions, like citing personal communication with permission, but generally peer-reviewed journals are the standard.
So Elena's pediatrician is almost certainly drawing on that body of peer-reviewed consensus science.
Overwhelmingly likely, yes. Whereas the online article, well, it might not meet those standards at all. It could be presenting a viewpoint that's way outside the mainstream scientific understanding, maybe even based on flawed or misrepresented information.
Okay, this is helpful. Let's dig a bit deeper into how scientists build that understanding. Experiments and observations.
Right. This is the engine room, really. Experiments are designed specifically to test hypotheses. You try to isolate one factor.
The independent variable.
That's the one, the thing you manipulate or change. And you see how it affects another factor.
The dependent variable. The outcome you may have.
Exactly. And crucially, you try to keep everything else the same. Those are your control variables, so you can be more confident that any change you see is actually due to the independent variable.
The material had that cool historical example, Lind and scurvy.
Ah, yes. James Lind, mid-1700s. Sailors were dying horribly from scurvy. He took a group of 6 sailors, divided them up.
Small groups, right?
Very small by modern standards, yeah. Only 12 men total. But he gave each pair a different supplement to their standard diet: cider, vinegar, seawater, sulfuric acid.
Ouch.
Yeah. And importantly, oranges and lemons for one pair.
The independent variable was the supplement.
Correct. And the dependent variable was, did they get better?
And the citrus group did.
Dramatically so. Even though they had no idea about vitamin C back then. It just shows how a structured experiment, even a simple one, can test an idea and lead to a breakthrough.
But you mentioned the small sample size. That matters, doesn't it?
Hugely. When you study only a few individuals, you run into issues of sampling. You're looking at a small sample, hoping it represents the larger group or population.
And if it's too small?
You might get results just due to random chance error. Or if your sample isn't representative, say, you only picked the healthiest sailors to begin with, you could have bias.
So bigger samples are better. And random selection.
Generally, yes. Larger, randomly selected samples give you much more confidence that your results aren't just a fluke and actually reflect the broader reality.
Like the polio vaccine trial the sources mentioned. Jonas Salk.
Oh, absolutely. A landmark example.
Yeah.
Mid-1950s, over 1 million children involved. Talk about reducing chance error.
Wow. And they had different groups.
Yes. A massive experimental group got the actual Salk vaccine, and a control group got a placebo.
A placebo. That's like a sugar pill, right?
Ugh.
But in this case, an injection.
Exactly. A harmless saline injection that looked identical to the vaccine. It had no medical effect itself.
Why use a placebo?
Yeah.
Why not just compare vaccinated kids to unvaccinated ones?
To control for the placebo effect. Sometimes people feel better or report improvement just because they believe they're getting a helpful treatment, even if it's inert.
The power of the mind.
It's a real phenomenon. Using a placebo helps ensure any difference seen between the groups is due to the vaccine itself, not just the expectation of getting treated.
And the Salk trial was also double-blind.
Yes, even better. Blind means the participants don't know if they're getting the real treatment or the placebo. Double-blind means neither the participants nor the researchers interacting with them know who is in which group until the study is over.
Why is that important?
It removes potential bias from both sides. Participants can't subconsciously report feeling better if they know they got the real thing, and researchers can't subconsciously influence outcomes or data recording based on knowing who got the vaccine. It's the gold standard for clinical trials today.
Okay, but what if you can't do an experiment? The sources mentioned ethics, practicality.
Right. You can't ethically assign people to smoke 2 packs a day to see if they get cancer. Sometimes manipulation just isn't feasible or allowed.
So then what?
Then scientists rely on observational studies.
It's just watching.
Essentially, yes.
Yeah.
Measuring characteristics, looking for patterns, but without intervening or manipulating variables. Political polls are a simple kind of observational study. You just ask people their opinion.
And the material outlined a few types.
Yeah, 3 main ones. First, cross-sectional studies. You collect data from a group at one single point in time, like surveying a population today about their smoking habits and whether they have lung cancer. Early links were found this way.
Okay.
Second, case control studies. These are retrospective. You find a group of people who have a condition, the cases, say lung cancer patients, and compare them to a similar group who don't have it, the controls. Then you look back in time, maybe through records or interviews, to see if certain factors like smoking were more common in the case group.
Like the Dahl and Hill study mentioned.
Exactly. Their early work on smoking and cancer used a case control design. The weakness, though, is relying on people's memories of the past, which can be fuzzy or biased.
Right. And the third type?
Cohort studies. These are perspective. You identify a group of people, the cohort, maybe people who are currently healthy, and you follow them forward over time, collecting data periodically.
So you see who develops the condition later.
Precisely. Dahl and Hill also did a famous cohort study following British doctors for 50 years, tracking their smoking habits and health outcomes.
50 years. Wow.
Yeah. Cohort studies can be very powerful because you're collecting data directly as things happen, which is usually more accurate than relying on past recall. But they take a long time and can be expensive.
Now, the sources really hammered this point. Correlation versus causation, especially with observational studies.
Absolutely critical distinction. Observational studies are great at finding correlations, associations between two things. Smokers have higher rates of lung cancer, that's a correlation.
But it doesn't automatically prove smoking causes cancer, not just from that observation.
Not definitively, no. There could theoretically be some other hidden factor, a confounding variable, that makes people both more likely to smoke and more likely to get cancer.
Like the example of ice cream sales and crime rates both going up in summer.
Perfect example. They're correlated, but ice cream doesn't cause crime. The heat is the likely confounding factor influencing both.
So experiments where you control variables are better for proving causation.
Much better, yes. By isolating the independent variable and using things like placebos and blinding, experiments give stronger evidence for a cause and effect link.
But we still do observational studies
But
because
sometimes experiments aren't possible, like we said, or observational studies might be cheaper, easier, or allow researchers to look at huge populations and find potential links that can then be investigated further, maybe with experiments if possible.
the sources also mention model organisms, like mice.
Right. When direct human experimentation is too risky, unethical, or just impossible, scientists often study biological processes in other species' model organisms that share relevant biology with humans, like using mice to study obesity or test potential drugs.
Okay, that makes sense. So let's tackle Elena's third question. Why are diseases like measles, mumps, polio so rare now? And if they're rare, why do we still need vaccines?
Yeah, it's a common question, and the answer really lies in the history and the incredible success of vaccination itself.
It goes back quite a way, doesn't it? Edward Jenner.
All the way back to 1796, Jenner, an English doctor, noticed something interesting. Milkmaids who caught cowpox, which was a pretty mild disease from cows.
They didn't seem to get smallpox, which was deadly.
Exactly. So he had this hypothesis. Maybe cowpox infection protects against smallpox. And he did a famous experiment.
On a boy, right?
Yeah, he took material from a cowpox sore on a milkmaid and inoculated an 8-year-old boy, James Phipps. The boy got mild cowpox. Then later, Jenner exposed the boy to smallpox material.
Risky.
Hugely risky by today's standards. But the boy didn't get sick. That was essentially the world's first vaccine using a related, milder disease to provide immunity.
And then Pasteur came along later.
Yes, Louis Pasteur in the late 1800s. His work on germ theory, understanding that specific microbes cause specific diseases, really revolutionized things and paved the way for developing more vaccines against diseases like rabies and anthrax.
The material also mentioned something even earlier, variolation.
Ah, yeah. That was practiced for centuries before Jenner, especially in Asia and Africa. It involved taking actual smallpox scabs, grinding them up, and sometimes blowing the powder into someone's nostrils or scratching it into their skin.
Seriously.
Yes. The idea was to cause a milder infection than naturally acquired smallpox, hoping to induce immunity. It was riskier than vaccination. People could still get very sick or die, but it was better than taking your chances with a full-blown smallpox epidemic. It shows people intuitively understood the concept of immunity for a long time.
So fast forward. How effective have vaccines been?
Phenomenally effective. Just look at measles. The source material highlighted this. In the U.S., thanks to widespread MMR vaccination, measles was declared eliminated, meaning no continuous transmission back in 2000.
Eliminated. But we still see outbreaks sometimes.
We do, unfortunately, usually when vaccination rates drop in certain communities. Globally, measles deaths plummeted by huge percentages after the vaccine became widely available. Smallpox was completely eradicated worldwide by 1980, a monumental achievement solely due to vaccination. Polio is almost eradicated.
So the answer to Elena's question is?
The reason these diseases are rare is because of vaccines. And the reason we still need to vaccinate is to keep them rare. If vaccination stops or falters, these diseases, which still exist in parts of the world, can come back fast.
It's about maintaining that herd immunity or community protection.
Exactly. When enough people are vaccinated, the disease can't spread easily, which protects everyone, including those who can't be vaccinated for medical reasons, like infants or people with weak immune systems.
Okay, that's clear. The sources touch briefly on analyzing data after a study.
Yeah, just that collecting data is only the first step. You have to organize it, analyze it, interpret it. That progresses from, simple notebooks in school.
Tables, graphs, maybe scatter plots to look for correlations in more advanced work.
Right. And always remembering that correlation doesn't equal causation when you interpret those graphs.
Gotcha. And statistics become important too.
Definitely. Especially in high school and beyond, using math and statistical tools helps evaluate the strength of relationships in the data, the likelihood that findings are real and not just chance.
And finally, the material emphasized good experimental design, getting it right from the start.
Absolutely crucial. If the design is flawed, the results might be meaningless no matter how fancy your analysis is. You need clear variables, proper controls.
And replicability, being able to repeat it.
Yes. Other scientists need to be able to replicate your experiment and hopefully get similar results. That builds confidence. The sources mention different ways to set up experiments.
Like independent measures,
Yeah.
different people in each group.
Right. Or repeated measures, where the same people experience all the different conditions or treatments.
But that could have order effects, couldn't it? like getting better at a task just by doing it twice.
It could, yes. So researchers use techniques like counterbalancing, changing the order of conditions for different participants to try and even that out.
And match pairs.
That's where you try to match participants in different groups on key characteristics like age or gender to make the groups as similar as possible before the experiment starts. Randomly assigning people to groups in independent measures designs also helps achieve similarity.
It also mentioned pre-specifying things, like planning the analysis beforehand.
Yes, especially in high-stakes research like clinical trials for drugs. Researchers often write a detailed statistical analysis plan, or SAP, before the trial begins. It outlines exactly how they intend to analyze the data.
Why do that?
It helps prevent cherry-picking results later, only reporting analyses that happen to look good. It increases objectivity. The experimental design itself actually dictates what statistical methods are appropriate.
So you need to know the design to pick the right stats test.
Exactly. And if researchers have to deviate from their original plan for some reason during the study, they need to communicate that clearly. It's all about transparency and rigor.
Okay, so let's try and tie all this back together for Elena and for everyone listening.
Right. We've seen that science isn't just a collection of facts. It's a dynamic process. It's about asking questions, developing ideas you can actually test,
Hypotheses that are falsifiable.
using careful methods like experiments or well-designed observational studies, analyzing the evidence critically, and being open to revising ideas when new evidence comes along.
And when we're faced with conflicting information, like Elena is, we need to think critically about the source.
Definitely. Is it based on solid peer-reviewed research? Does it represent scientific consensus? Or is it an outlier claim?
What kind of evidence is presented? Is it just anecdotes or correlations from observational studies or stronger causal evidence from experiments?
And importantly, is it acknowledging the difference between correlation and causation? Are there potential biases or alternative explanations being ignored?
So for Elena, her pediatrician's advice about the MMR vaccine, that's rooted in decades of consensus science.
Yes, built on countless large-scale, peer-reviewed studies involving millions of children worldwide, consistently showing the vaccine is safe and highly effective at preventing serious diseases.
Whereas the online article claiming a link to autism.
That stems from discredited research and goes against the overwhelming scientific evidence. The rarity of diseases like measles today is actually powerful proof of vaccine effectiveness, not a reason to stop vaccinating.
It really shows how science, when done rigorously, can lead to huge public health benefits.
Absolutely. Vaccination is one of the greatest public health success stories in history.
So maybe a final thought to leave people with. In this age of information overload, how do we cultivate that inner scientist?
That's a great way to put it.
How do we all get better at asking those critical questions, seeking out reliable evidence, maybe being a bit more skeptical in a healthy way about information we encounter everywhere, not just about science?
Yeah, it's about developing those critical thinking muscles, understanding the process of how we come to know things reliably. That understanding is really empowering, isn't it? It helps us navigate the noise.
It really does. Well, thanks for taking this deep dive with us today.